welcome back to Crack Classroom. Today this is going to be history, I hope. We're going to try to get through this chapter as quickly as possible because it's B1, and B1 is the quickest chapter to ever grace biology. Even though it does have 10 parts of the chapter, I fit it all onto two sides of A4 with a tiny bit on another piece. But we're going to try to get through this as quickly as possible. Now, we're going to start with a microscope. And microscope questions are usually on those little B1 tests that we did in like year 7, but you never know what you're going to get because tests are kind of like a box of chocolates. You never know what you're going to get. Forrest Gump said it, I'm repeating it. Now here you have a microscope and you have the eyepiece at the top which you look through. Then you have here you have the objective lens, which you can adjust the magnification, which usually goes from 10 to 20 to 40. Then you have the stage, which is kind of like a black stage with a little hole that lets the light through, because this is a light microscope, not an electron microscope, because the light microscopes are cheaper. Then on the stage you have the stage clip, which keeps your slide in place, and your slide usually has the thing that you're looking at in it. Then you have the coarse adjustment knob, or you can call it the coarse focus knob, or the coarse refinement knob. Not coarse refinement knob, just coarse adjustment knob or coarse focus knob. Then below it you have the fine focus knob or the fine adjustment knob, whichever way you want to call it. So you have light microscopes and you have electron microscopes. All you need to remember is that you can have two kinds. Light microscopes are cheaper, electron microscopes are expensive. Electron microscopes are ginormous, light microscopes are small and portable. Light microscopes go up to 2000 magnification, whereas electron microscopes go up to 200,000 magnification. I can't wait to use one of those, maybe in the future. That would be really fun. It's just wow, science. Anyway, you have to remember this calculation, and it's called I am. I remember it as I am because I am a piece of shit. I'm making myself laugh. But you write it as I at the top, then A and M. You can write M and A any way you want. I know you know that, you know math. Anyway, I stands for image size, A stands for actual size, and M stands for magnification. But when you're doing this, you have to convert millimeters to micrometers. And micrometers is the weird long M. So 1 millimeter is 1,000 micrometers. So obviously if you're going from millimeters to micrometers, you're going to times it by 1,000. And if you're going from micrometers to millimeters, which I don't even know why you'd be doing that, but maybe the question asks it, you divide by 1,000. Good? Good. Now, moving on to our little cells. So let's start with the animal cell. That's the easiest one. Let me just draw this really quickly. You don't need to be very specific about this as long as you can tell what it is. I don't really care. Okay, we have a very simple drawing. I'm going to colour the nucleus black so you can see it. We have a very simple animal cell. So the black thing is a nucleus. These little dots are the mitochondria, and this thing that's encompassing it is called the cell membrane. The thing inside, all of the stuff that it's floating in, is called the cytoplasm, and then this little thing is the ribosome. Or sometimes the ribosomes are depicted as like smaller things, but usually the ribosome is depicted as like a clump. But we'll later find out. Well, you're not going to do A-level biology, but ribosomes don't look like that, obviously. Ribosomes are the little things that... whatever. But after I explain a plant cell, then I'll explain what all of these things' functions are. So I'm just going to draw a lovely little plant cell right now. Lovely. So the black thing, of course, is the nucleus. Then you have the outside wall, which is the cell wall. Then you have the inside, and it's the same as the animal cell, and that's called the cell membrane. Then you have this large thing, that is the permanent vacuole. You have to call it permanent. Don't know why, don't ask me. This thing in the corner, same thing as that, it's the ribosomes. Then you have these bigger things. Now these bigger things are chloroplasts, and the black dots, again, are mitochondria. So now, you're probably wondering, what do all these things do, Freddie? Well, I can tell you, Anna. The nucleus controls what is happening inside the cell, but it also contains the genetic material and chromosomes. Then the cell wall, that controls what moves in and out of the cell. Then you have the cell wall, which basically just maintains the structure of the cell. Then you have the mitochondria, which is where respiration takes place. And then you have the cytoplasm, where gas exchange takes place and everything just floats in it. Then you have ribosomes, where protein synthesis takes place. Chloroplasts, where chlorophyll is stored, and chlorophyll, of course, converts chlorophyll is green, and it converts sunlight. It absorbs sunlight for the plant to use in photosynthesis to make food. Then you have the permanent vacuole, which is cell sap, and it keeps the cell rigid. And you have the eukaryotic cell, and now we're moving on. We're done with those. Those were our eukaryotic cells, and our eukaryotic cells are usually animal cells and plant cells. Fuck. I'm losing my voice. I've recorded so many of these. Eukaryotic cells have a cell membrane, they have cytoplasm, and they have genetic material enclosed in a nucleus. Those are the three key features of a eukaryotic cell. Now we go on to a prokaryotic cell. A prokaryotic cell is usually bacteria or bacterium, whichever way you want to say it. Don't care. Good for you. Bacteria, instead of having a nucleus, they have small rings of DNA called plasmids. Now, we're going on to our prokaryotic cell. I'm just going to draw a very basic bacteria. Now, a bacteria, unlike our little plant and animal cells, has 3 layers, kind of like an onion. Anyway. Okay. This is our prokaryotic cell, or bacterium. As you can see, it has these little tails. You'll remember those from a sperm cell. These are plural, flagellum, or flagella. Flagella is the plural, but singular is flagellum. Flagella is spelled F-L-A-G-E-L-L-A, flagella. Then you have these 2 little rings there, those are plasmids. Then you have this, and this is just a big strand of DNA. It's free floating, remember, it's not a nucleus. Then you have this outside wall, and that is the slime capsule. It's not present in all bacterium, but it's present in most. Then you go in further, and you have the cell wall, and then furthermore, cell membrane. Good? Good. Okay, nerve cell. Nerve cells are a bit more complicated. I'm not going to draw it now because it's a bit too hard and I don't want to waste time because this is going to be the quickest episode. So a nerve cell looks like this. And in the centre you have the nucleus, of course. Then you just have the cell body. These little lovely things are dendrites. So these are dendrites. Then you have this little encasing of the axon. The axon is like the thing that runs through it all, but the things encasing it that kind of look like pencil grips, those are I call it the myelin sheath, but some people call it myelin. It's spelled M-Y-E-L-I-N, so whichever way you want to pronounce it, but it's the myelin sheath. The axon then connects into here, and as you can see, there's more things, but these aren't called dendrites this time. Those are called synapses, and we'll get into those later in another chapter where we talk about synapses and gas exchange. But this nerve cell has many dendrites, so remember, these little things on the cell body, the things at the top of our little, it kind of looks like a pipe cleaner. Remember, it's a pipe cleaner that you made in like primary school, but these little dendrites are for connection to other cells. The axon, so that long thing that spans throughout it, kind of looking like a spaghetti. The axon sends nerve impulses from one place to another, and it can be very long. Our longest example of an axon is from the top of our spine to the base of our spine. It is literally our spinal cord. Then we have the synapses, the things at the bottom, kind of like a broom. Okay, kind of like a broom. The synapses are adapted to pass impulses to another cell. That's what we'll get later into. Or between a nerve cell and a muscle. We'll get into that later in another chapter. Then you have a muscle cells, which kind of looks like an evil eye, but it's really weird. Okay, I don't think you're ever going to get a muscle cell in an exam, but if you do, I'm going to give you some little adaptations of the muscle cell. So a muscle cell has a lot of mitochondria for energy, because, of course, if we're moving our muscles a lot, they're going to need a lot of mitochondria for respiration, because respiration, a byproduct of respiration, is energy. That's why there's a bunch of mitochondria there. And relevance, we've just learnt this in biology, but if you're watching this at a later time, then you haven't just learnt this in biology. But muscle cells can store glycogen, or glycogen, spelled Gly. Glykogene, glycogen, or I say glycogen, you can say glycogen, whichever way you want to say it, whichever way helps you remember it. Yeah, so that's what the muscle cells do. Then we go on to another specialised cell, which is a sperm cell, and sperm cell has a large nucleus, then it has the acrosome, acrosome, I always say that wrong. Then we have the midsection, which contains a lot of mitochondria, and then we have the tail, or the flagella, because it's singular. The flagella, or the tail, helps them to swim. Then you have the midsection of the sperm cell, which contains a lot of mitochondria. Again, mitochondria is where respiration takes place. A byproduct of respiration is energy. And what does a sperm cell need? A lot of energy, because it's swimming through your vagina. Remember, sperm is placed in the vagina at 28 miles per hour. Thanks, Miss Fotheringham, you pregnant bitch. Anyway, sorry. Then you have the acrosome, which is at the front, the head of the sperm. Just remember my head like this. The acrosome stores a digestive enzyme, which breaks through the little lovely shell of our eggs. The nucleus contains genetic information, of course, because if it is making a baby, then it's going to need some genetic information from the father. Then we get onto a root hair cell, which I always remember because it kind of looks like an old man. See, eye, ear, long nose. Anyway, at top we have the nucleus. Then we have a permanent vacuole again. We have a bunch of mitochondria, and we have a root hair. So our lovely little root hair cell has a large permanent vacuole, which speeds up water movement by osmosis, because of course osmosis is occurring. It increases surface area, the long like root hair. It increases surface area for water to move into the cell, and it has many mitochondria, because respiration, energy, remember, which is needed for active transport, because remember, active transport is going against the concentration gradient, so it needs a lot of energy to occur, and to get that energy you need a bunch of fucking mitochondria. Xylem cells. We have our xylem cells. Our xylem cells are the ones that are dead, and it only goes in one direction, from root to shoot. And it has a bunch of lignin spirals, because remember, it generates lignin. It's a hollow tube, and it's fucking dead. Okay? Then we have a phloem. Our phloem, remember, is surrounded by companion cells. Remember the stupid thing that I said doesn't sound very scientific, because it's just another word for friend. So it has companion cells, which have a bunch of mitochondria, which provide energy for active transport. Phloem, in contrasting what happens in the xylem, it goes two ways. It goes root to shoot, and it also goes shoot to root. Both ways. It can't decide. It's bisexual. Anyway, diffusion. Diffusion is going with the concentration gradient. It goes from a high concentration to a high concentration to a low concentration. So you to me. Okay? High concentration to a low concentration of particles. If there is a big difference in concentration, then it happens very quickly. If there is a small difference in concentration, then it happens very slowly, because it doesn't really care much. So another calculation, net movement. Net movement is particles moving in minus particles moving out. That is net movement. That is your calculation. Then temperature also affects diffusion. If there is a high temperature, then diffusion happens quickly. If there is a low temperature, diffusion is very slow. It's like, no, I'm tired. It's me on a winter's day. It's you on a winter's day. You every day. You're very cold. Quick coffee break, right back. Osmosis. Okay, it also moves with the concentration gradient. So it's a movement of water molecules from a high concentration to a low concentration of water molecules through a partially permeable membrane. Remember, partially permeable membrane, or you can say semi-permeable, whichever, but I'd rather go for partially permeable membrane, because remember, biology mark schemes are very specific, so you'd want to remember partially permeable membrane. Okay, in our blood cells in an animal, you have isotonic, which looks like this. Isotonic is kind of like equilibrium, where there is the concentration of inside the cell and outside the cell are the same, which makes it isotonic. So basically, water goes in, water goes out. Makes it even. It's lovely and circular. But sometimes our cells get a bit too greedy and they take in too much water. So there is a higher concentration of water inside the cell than outside the cell. And it causes it to burst, because our poor little blood cells, remember, because our animal cells do not have a cell wall. The cell wall maintains the structure of the cell, but we do not have those. We only have our puny little cell membrane, so it bursts if there is too much water. So too much water is going in, it's called hypotonic. Hypotonic. I would have called it hypertonic, but it's hypo. Hypo, spelled Hypotonic. Hypotonic. Then we get onto hypertonic, which is where the concentration of water outside the cell is larger than the concentration inside the cell. So our blood cell is being too generous and it's giving too much water outside, and our cell kind of like shrivels up and dies. Then you have a plant cell. Now, a plant cell is structurally sound and it has a cell wall to support itself, because it has support, because us humans will have a breakdown, kind of like Freya, but plants, they're just living life, okay? They have a cell wall to support them at all times. So when they are getting too much water, they just become turgid. They're just like, hey, I'm just not going to let any more water in. So the pressure builds up till no more water can get into the cell. And they're fine. By the way, turgid looks like this. Then we have our little equilibrium stage, which is called flaccid, where enough water is coming in and enough water is coming out. Then we have plasmolysed, which is spelled PLASMOLYSED, where too much water is going out. But instead of shriveling up and dying, since they have the cell wall to maintain the structure of the plant cell, the cytoplasm just kind of like stretches off the cell and kind of looks like an anorexic person, like Dahlia on a climbing frame. Okay, then you have the last one of them, active transport. Active transport goes against the concentration gradient, so it's going from a low concentration to a high concentration. And obviously, if you were climbing Mount Everest, you would need a lot of energy. So remember, active transport is kind of like climbing Mount Everest. They need a lot of energy. So usually when a cell, or in a plant, or in a human, if active transport is occurring, they're going to need a lot of mitochondria for a lot of energy. So a lot of energy is needed to carry out active transport. Then we have surface area to volume ratio. Surface area to volume ratio is rho SA to V. I think you know how ratio works. Anyway, small objects have a large surface area to volume ratio. Big objects have a small surface area to volume ratio, which makes exchanging materials quickly very difficult. But we have some adaptations. Some adaptations include thin membranes, which makes a short diffusion path, efficient blood supplies, which moves diffusing substances away from the exchange area to maintain a steep concentration gradient. So, for example, in plants, they have flat, thin leaves, They have air spaces in the leaf tissue and stomata, which make a big surface area and maintain a steep concentration gradient. Then in people, we have our alveoles, remember, the ball sacks for the lungs. They have a big surface area, and they have a large network of capillaries surrounding them, and the cell wall is also 1 cell thick. Then we have our villi in our small intestine, which has a large surface area because it's all like clumped together, remember? Kind of like this. Kind of like a fabric. Anyway, it has a short diffusion path, and it has a network of capillaries. And that is time. We are completely done with that. That was only 20 minutes. Some of the other ones are 40 minutes long. Wasn't that great? Anyway, I'm going to end this before it gets 21 minutes. Have a nice time studying biology. Hope that helped.